Hello, my name is Leo Ivey, and I am a junior biology student working with Dr. Bryan McLean. My project is entitled Testing Dehnel's Phenomena in Small Mammals of the Eastern United States. So, what is Dehnel's phenomena? Simply put, Dehnel's phenomena is a seasonal loss and regrowth of the brain as a method of energy conservation when resources are scarce. This phenomena, which was first described in 1949, has been observed in red-toothed shrews, weasels, mink, as well as moles. The intensity of the winter skull flattening and subsequent spring regrowth is dependent on environmental conditions. That is, it is less noticeable in populations that experience milder winters. It is also thought to be associated with certain characteristics of an organism's life, particularly high activity levels in winter. This phenomena has been extensively studied in Europe and Asia, but has been scarcely looked at in North American small mammal species. For this project, we have decided to focus on three main species of small mammals. These are the North American deer mouse, Peromyscus maniculatus, and two Appalachian shrew species, the masked shrew, Sorex cinereus, and the smoky shrew, Sorex phenius. There is conflicting evidence supporting the presence of the phenomena outside of shrew species, particularly in rodents, which includes mice. However, these mice exhibit characteristics that we usually associate with the phenomena, primarily high activity levels even in the winter months. The two shrew species are both in the genus Sorex, which has extensive literature support for other species in this genus. That being said, these two species have not been thoroughly examined for their phenomena previously. As a result of this, we predict that both cinereus and phenius will exhibit morphological changes in the skull that are in line with the documented effects of the phenomena. However, due to very little supporting evidence in other rodent species, we do not expect to see the same morphological changes in mice. Additionally, any seasonal changes in skull morphology may be difficult to distinguish in mice due to their longer lifespans and more complex population structures. All specimens collected will be CT scanned in order to examine the effects of the phenomena, and measurements of multiple skull proportions will be taken as our primary measure of the phenomena, using protocols established by Lazaro et al. Scans will be measured using the ImageJ software using the BoneJ plugin. Age will be determined based on tooth wear, and sex will be established based on presence of internal reproductive organs. Collected measurements will be tested with generalized linear models to test the effects of age, sex, and season on skull proportion. Additionally, species-specific principal component analyses will be used to visualize specimens in morphological space. Our preliminary focus has been on field collection for each of the three species. All individuals were live caught and euthanized in the field and have been preserved in ethanol. All Peromyscus maniculatus specimens were collected monthly last year using Sherman traps, giving us a total of 114 specimens. Shrew sampling has been ongoing since May 2021 and will continue through the end of the year. 4 more sampling sessions will be conducted targeting winter shrews, as well as fall and spring phenius specifically. Currently, we have 67 cinereus individuals and 19 phenius individuals. Shrews are targeted using a mix of pitfall traps, as well as Longworth and non-folding small Sherman traps. Finally, I would like to thank my mentor, Dr. Bryan McLean, as well as the other members of the McLean lab. Additionally, I would like to specifically thank Dr. Dehnel for her extensive advice and methodology, as well as the UNCG Undergraduate Research and Creativity Office, the STAMPS program, and the biology department for their financial support of this project.